Hello and welcome everyone. In this podcast we're going to talk about eukaryotic gene regulation as it relates to alternative splicing and RNA stability. But let's start off with alternative splicing. We have talked about this a little bit already, but let's fine-tune some of that discussion. And remember, we said you could have a gene that had multiple exons. In this example, we have 7 exons, and in between them we have introns. And when we first introduced this idea, we said that all of these introns are removed. And in doing so, you end up with a mature messenger RNA that would contain a message with 1 through 7 exon. So 1, 2, 3, 4, 5, 6, 7. Okay, so it would have all 7 exons. We alluded to the fact that sometimes we don't always get all 7, because we mentioned the case with the fruitless gene in Drosophila, how this is true in the males, but it's not true in the females. In the females, they end up splicing out a large portion of this. And though the fruitless gene doesn't have 7 exons like we have in this example, we can use it to kind of explain the general principle of it. And so maybe in the fruitless gene, we end up with only 1 and 7 exons. We miss all these ones in the middle. And the way this happens is that we talked about the splicing. When you had this 5' and 3' splice site for each of these introns. I should draw them closer to the exons here, like so. Now, these splice sites are usually used to remove each intron independently. What can happen though is that spliceosome that comes along, it can cut this 5' splice site, and remember we said usually then that will loop around and attach to a branch point here in this intron. But sometimes once you've cut this, it could loop around with a different intron. And in this case that we just talked about here, it's going to loop around with the branch point of the intron between 6 and 7. If this were to happen, you would end up with a situation, and I'm going to erase this here for a moment to give us some room to draw. What happens here then is you end up with this exon 1 being held by that spliceosome, and then you have exon 7 here also being held by the same spliceosome so they don't fall away from each other. And then you end up with this really large lariat. And this large lariat will contain an intron, then exon 2, then an intron, then exon 3, and then an intron, and then exon 4, and then an intron, and exon 5. We're going to have to make this a little bigger. Then an intron, and exon 6. And now 1 and 7 are the only ones not part of this, what we call a lariat. And as we mentioned before, this lariat will be degraded. So that gets taken care of. And then the spliceosome seals the deal now by merging these 2 exons together. And you end up with a mature messenger RNA, in this case here with only exon 1 and 7. And so this is how the fruitless gene is alternatively spliced, by removing a large portion of the middle exons out of it. And so now when this is expressed or translated into a protein, not only is it a completely different protein, but it's a completely non-functional protein, which is what we see in the female Drosophila, where it doesn't exhibit the mating ritual of the male. And the male has that gene, so it does have that mating ritual. So I want to talk about one more example of alternative splicing. With the fruitless gene, that explained how you could get 2 different proteins from the same gene depending on if you're male or female. But that doesn't explain this problem we have, where that within, say, humans, we may have greater than 200,000 proteins. So there are two ways you could make 200,000 proteins. You could either have choice one, you could have 200,000 genes. Or you could have fewer than 200,000 genes. For now, we'll just write less than 200,000 genes. If you had 200,000 genes, meaning one gene for each protein made, that could work. The problem is that throughout the course of evolution, what would have had to have happened was that new genes would have had to have evolved for each one of these new proteins that were being made. A more efficient way would be to have less than 200,000 genes and be able to take those existing genes that used to make just one protein, but now allow that existing gene to make 2, 3, 4, 5, or more proteins from that same gene. That would be much more efficient. That way, throughout the course of evolution, you wouldn't have to have new genes be developed or have them evolve from pre-existing genes. You could take that same gene and somehow or another express it differently to give you different proteins. And that's where alternative splicing comes in, because we know that in humans, for instance, we have between, we'll say around 22,000 genes. There are some estimates that are a little lower than that, and some that are higher, but we can go with 22,000 genes. These 22,000 genes, approximately, will make approximately 200,000 proteins. So the only way that you can do this is by using the genes in multiple ways. And that's where alternative splicing comes in. So let's talk about, just real quickly, this example of tropomyosin. There are other examples as well, but we'll just mention this one, tropomyosin. Particularly, let's talk about alpha tropomyosin. And this is a gene that's involved in the cytoskeleton. I don't know if you've all had cell biology yet, but if you've had, then you know that tropomyosin is very important in regulating the cytoskeleton. And you probably learned about it more as its association with muscles. This isn't a physiology class, so we're not going to talk about exactly what it does, but you should know that it's involved in the cytoskeleton and muscles. At least for this purpose of this discussion. I couldn't imagine asking an exam question asking whether or not tropomyosin was involved in the cytoskeleton. This is just an example to illustrate this important concept. The gene for tropomyosin ends up having 14 exons. So we go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and we're going to sneak one in here, 14. This tropomyosin, or atropomyosin enzyme, is important in several functions. It's important in striated muscle. It's important or found in smooth muscle. It's important in fibroblast cells. It's also important in brain cells, in neurons. Now, each of these 4 cells have this tropomyosin protein. And each of these 4 cells make that tropomyosin protein using this same gene. However, it is alternatively spliced in each of these. For instance, in the striated muscle cells, it contains exons 1, 3, 5 through 7, and 9 through 13. In the smooth muscle, it contains exon 1 and 2, exons 5 through 7, and exons 9 through 12, and 14. In the fibroblast cells, it contains 1 and 3, 5 through 7, 9 through 12, and then 14. In neurons, it contains 1 and 3, 5 through 7, 9 through 12, and that's it. The purpose of explaining this example isn't so that you can memorize all this. In fact, I don't want you to memorize what's specific about the tropomyosin gene in each of these cells. What I want you to know is that using this one gene with 14 exons, you can combine those 14 exons in many different ways to make a slightly different product. In this case, a slightly different tropomyosin protein that is specific to the needs of each of these individual cells. Evolution could have chosen to make 4 different genes, but that wasn't as efficient. It was better to take a pre-existing gene that essentially had the same things you needed in each of these cells with slight differences. So you can see between striated and smooth muscles, very similar, except we're exchanging exon 2 and 3 and 13 and 14. It's much easier to do this than to make a brand new gene. Just adjust the one you already have. Okay, let's go ahead and erase this and talk a little bit about RNA stability. Okay, RNA stability. And I mainly want to talk about these small interfering RNA molecules, which we're going to call lowercase s, lowercase I, RNA, siRNA. And all I want to mention about these is, remember that in a eukaryotic cell, we have our nucleus here, and this nucleus will make a messenger RNA through transcription. It'll be processed in all the ways that we learn, and then it leaves the cell. And then we can have these small interfering RNAs in the cell. Now, a lot of times these small interfering RNAs will come in through a virus, and this is the way a virus can come in and shut down the expression of certain genes and either kill the cell or affect the overall ability of that virus to survive within the cell. So let's have a virus out here. That brings in this siRNA. We'll draw it like this. And what this small interfering RNA can do is it will bind to the RNA that was just made. And so this interfering RNA would have to have the reverse complement to it, so it can bind in an anti-parallel manner. And then I'm going to leave out a few steps here, but ultimately what happens is this messenger RNA will be degraded. So this small interfering RNA can come into the cell, bind with the messenger RNA, and then targets it for destruction, and it's degraded. Okay, let's have a short summary here. We talked about 2 main things in this podcast. We talked about alternative splicing and short interfering RNAs. With alternative splicing, we talked about how we can take one gene with many exons and create several proteins. So one gene, using alternative splicing, we can make different proteins. And how, evolutionarily speaking, this is more advantageous than making 3 genes to make 3 proteins. We then talked about these small interfering RNAs and how they can come into the cell by the source of a virus and alter the expression of certain genes within the whole cell. Okay, that's all I have for this podcast. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.